Okay. All right. I think we can get started in the interest of time. It's so good to see everybody for this UCLA Cannabis Research Initiative special seminar with Dr. Elisa Pabon. Just a couple of announcements before we start. The first announcement, again, is to remind everybody about the annual Center for Medicinal Cannabis Research Symposium that's happening on April 21st and April 22nd. That's Thursday and Friday of next month. And we've passed along links to register for that. So we'll pass along a link again. We're supposed to have our faculty meeting, our monthly faculty meeting that Thursday, April 21st. But in the interest of supporting CMCR, the Center for Medicinal Cannabis Research, and I know people are going to want to attend that anyway, we're going to move our faculty meeting to April 28th from 12 to 1. So we'll send out a reminder about that. And we got a lot of RSVPs for this special seminar. We know that not everybody can attend right now, so we are recording this event and we're going to post it on our website. Please shoot me a text or not a text, shoot us an email if you want the link for the recording. We also recorded the Cannabis Grand Rounds with Dr. George that happened 2 weeks ago. So that link, I think it's available. Christina, is that available on our website yet?
Yes, it is. I can also place it in the chat box as well.
Okay, great. So that link is available on the chat box. So now to get to our event today, it is a pleasure to welcome Dr. Elisa Pabon. She is a postdoctoral scholar who completed her doctoral fellowship in neurobiology at the University of Chicago. Her work in human psychopharmacology began as an undergraduate student at the University of Pittsburgh, where she worked with Casey Creswell and Lynn Reeder. And across the past 7 years of her journey through human behavioral research, Lisa's project spanned from investigating self-report verbal biases in chronic cigarette smokers to brain imaging studies of how neural activation during non-drug rewards relate to behavioral responses to alcohol. Her current interest and expertise and specialization is in cannabinoid research, which began with developing a phone-based measure of oral THC impairment. Witnessing individual variability in responses to THC left her eager to uncover neurobiological sources of variation, which led her to her dissertation research, which found evidence of hormone-related variability in female responses to THC across the follicular phase of the menstrual cycle. Today, she'll be talking to us about this interest, investigating sex and hormone-related differences in responses to cannabinoids and other psychoactive substances. Dr. Pabon has received several awards from numerous societies, including ICRS, the International Cannabinoid Research Society, Experimental Biology, Scientista Women in STEM Conference, the annual Biomedical Research Conference for Minority Students, and of course, CPDD, College on Problems of Drug Dependence, CPDD. So I first met Elisa back in 2019. It feels like yesterday, but I can't believe it's almost 3 years ago. She reached out to me about a symposium that she wanted to submit for the annual CPDD meeting. She seamlessly organized 6 junior and senior principal investigators, from preclinical researchers to human subject scientists, to provide a translational overview on the sex-dependent effects of cannabinoids that would capture the interests of preclinical researchers, clinical scientists, and epidemiologists. This is not an easy task to do, by the way. The panel was accepted, and although I'm a little biased, people shared with me that it was one of the best organized and content-filled panels of the meeting. So it's really a pleasure to welcome Dr. Pabon, and today she'll be talking to us on the topic of hormone-related variability in female responses to oral THC across the follicular phase. And I welcome attendees to put questions in the chat box, and after the seminar, we will be able to ask Dr. Pabon those questions. So thank you so much. Enjoy.
Thank you so much, Dr. Cooper. That introduction was incredibly generous and gracious. It is an absolute pleasure and honor to be invited to give this talk today, and I really look forward to speaking with all of you. I'm going to go ahead and share my screen so you can all see my first slide. One moment. that. Beautiful. Thumbs up that you guys can see my slide. Awesome, great. So as Dr. Cooper mentioned, today I'm going to tell you about my recent dissertation research. I graduated back in December 2021, and it was regarding hormone-related variability in female responses to oral delta-9 tetrahydrocannabinol, also known as THC, across the female follicular phase. Before I really dive into this, I want to tell you a little bit about myself. Obviously, Dr. Cooper gave an incredible introduction, but I wanted to give you a little bit more details. One thing that I find is very strongly identifiable for me is that I am mixed race. I'm half Colombian and then half Greek Italian as well. I'm bilingual, and I find it to be a huge part of my identity. During my Bachelor's of Science, in which I got a Bachelor's of Science in Biological Sciences and Psychology at Carnegie Mellon University, as Dr. Cooper mentioned, I had a variety of different research experiences, ranging from social psychology to fMRI to clinical behavioral health research. This all led me to develop a really deep passion in research and decided to pursue a PhD or doctoral degree in neurobiology at the University of Chicago, with the pleasure of working with Dr. Harriet DeWitt in her human behavioral pharmacology lab. And so to tell you a little bit more about why I chose to focus on substance use, well, there are a few things I find very interesting. One, substance use is prevalent in the human experience. Nearly every person on Earth uses some sort of psychoactive substance, whether it's caffeine, nicotine, alcohol, you name it. And that prevalence in the human experience is something that very much interests me. Why do so many people use substances? Another interest is the development and maintenance of substance use disorders. Why do some individuals progress from initial substance use to substance use disorders? The repeated use, the use despite negative consequences, what is that behavioral and neural pathway and journey they go on? And then lastly, something that ties into both of these is that individuals respond differently to different categories of drugs. And this is called individual variation, individual variability, individual differences. It's a huge area of substance use research, and it can tell us a bit more about why certain individuals are more likely to develop substance use disorders, or why some individuals are more likely to be averse to some substances. And all of these tie together really to blossom into one of my most recent passions, is my interest in drug policy and how the connection between basic science and clinical research tie into and justify drug policy. And unfortunately, how in the past, drug policy has not usually looked at research and tends to marginalize and target certain populations of individuals. So just a general background as to why I'm so passionate and interested in this field. Moving back to my dissertation research. Today, I'm going to talk to you a little bit about why I chose to study cannabis specifically out of all the substances of abuse, and then why I chose to focus specifically on females or women. I'll go over some pertinent and important preclinical evidence that really justified and founded why I chose to explore this question and topic in humans. This led me to develop two main research questions, which I'll go over, and with those questions, two main hypotheses. I'll then touch on the method I used to attempt to answer these questions. I'll go over the results, which I have multiple parts of results. First is just the ovarian hormone analysis, and then the menstrual cycle phase analysis. I'll go over the conclusions of these results and then implications in future directions. First, I'll start with my rationale. So some of you, maybe all of you, are familiar with cannabis, given this is a Cannabis Research Initiative seminar, but those who may not be, this is a graph of the various legal status of cannabis across the United States. Don't be mistaken, federally, cannabis remains a Schedule I controlled substance, yet each state has its own legislation on whether cannabis either is legalized medicinally, adult recreational use, or decriminalized only. And this rapidly changing policy across the nation is increasing the accessibility of the substance. So even though there hasn't been a lot of evidence that these changes in policy increase rates of use, the ease of accessibility is changing. Thus, there is a need for additional research to understand this plant and how it affects people, not only in the short term, but also in the long term. So what do we know so far about cannabis? Well, it can cause a variety of different effects. It can cause some positive effects, like it can be relaxing, it can be anxiety relieving or anxiolytic. It can also be stimulating to the appetite. It can cause hunger cravings and can be useful for individuals who tend to undereat or are going through chemotherapy and experience a lot of nausea. Additionally, there is evidence that it can help with pain or have pain relieving analgesic effects. Now, in addition to these more positive or potentially therapeutic benefits of cannabis, there are some more negative consequences. For example, tachycardia, or a heart rate above 100 for over an hour, tends to happen after use of smoked cannabis. And additionally, individuals tend to get quite paranoid, or certain individuals can get quite paranoid and scared after the use. And lastly, it can also be anxiogenic in some individuals, which is interesting since one of the first positive effects we listed was anxiety relieving, anxiolytic. And so this leads me into what do we know about where these effects come from? Well, it's known that THC, or delta-9 tetrahydrocannabinol, is one of the over 100 different identified cannabinoids within cannabis plant, and it binds to cannabinoid-specific receptors. It's believed that THC's partial agonist action at the CB1 receptor in the central nervous system is what specifically causes the effects of smoked marijuana, both physiologically and psychologically. So what we can take from all of this is that in cannabis, THC is the main psychoactive constituent. It's the one thing that's causing most of the subjective high that we get from anecdotal reports. And we can see in this figure relative density maps of cannabinoid type receptor 1 throughout the brain. So that's CB1 receptor density in different areas. And you can see in both figures that it's most highly found, or most densely found, in the cerebellum and a few of the limbic areas, but it's also found widespread throughout the cortex. And you can see some of the different regions it's found in and how they're all involved in a variety of different physiological functions. So it's no wonder that when smoked cannabis is consumed, or THC is consumed, it causes a variety of different effects in a variety of different individuals. So I've had the opportunity to work with THC in humans for a while now, and I've gained some knowledge on some of the regular or prototypical acute effects we see with THC administration. I was able to work on developing a phone-based measure of THC intoxication, and during this time, I really saw consistent increases in heart rate. I saw consistent cognitive impairment in both working memory and reaction time, and I saw consistent increases in subjective readings of feeling high, stoned, feeling euphoric, or feelings of well-being. In addition, sedation and fatigue, confusion, and anxiety. Some questions that came to my mind after seeing these results was this increase in heart rate due to the sympathetic nervous system activating or the parasympathetic nervous system activating, or could it have been both? And then another question, again, something I alluded to earlier, is what might predispose an individual to be more susceptible to the more positive effects of cannabis and THC versus the more anxiogenic or aversive effects of THC. So these are all the questions that led me to study cannabis and THC. Now, why did I choose to specifically focus on women? Well, first, unfortunately, women have been historically underrepresented in cannabis-related research. This is actually true in a lot of other clinical fields. In addition to that, research on women has shown that women are more susceptible to adverse responses to cannabis, as I mentioned, tachycardia, anxiety, paranoia. They also show faster progression from initial cannabis use to the development of cannabis use disorder, an effect called the telescoping effect. And also, women who are coming to treatment for cannabis use disorder exhibit more severe withdrawal symptoms and have a higher likelihood of relapse when compared to men seeking treatment for cannabis use disorder. Now, what might be underlying some of these things? Well, it's thought that ovarian hormones might actually cause these women-specific effects or sensitivity. And ovarian hormones may, in effect, affect responses to THC. So for those of you who might not be too familiar with the menstrual cycle or ovarian hormones, in front of you, you have a figure depicting the average menstrual cycle, approximately 28 days on the x-axis, and then relative hormone levels on the y-axis. Although this says 28 days as the average cycle length, there is a lot of variability between women, so take that with a grain of salt. What you can see is that the menstrual cycle phase has two major hormone fluctuations, estrogen and progesterone. Estrogen is the lighter blue, which peaks during the follicular phase, and progesterone is the darker blue, which peaks during the luteal phase. Now, you can break this cycle down into further subdivisions by looking again at follicular phase and luteal phase, and then you can subdivide it even more by early follicular phase, where you see relatively low estrogen levels, and late follicular phase, where you see relatively high estrogen levels. I chose to focus on the follicular phase because there is evidence that progesterone may have its own effects on certain physiological functions that may affect drug responses. So for my first journey into this field, I decided to focus my study on the follicular phase to isolate just the change in estrogen without interference from progesterone, or as much interference from progesterone. So now that I've given you a little background as to why I chose cannabis, why I chose women, I'm going to go into the preclinical evidence that really justified why I chose to do what I did. So firstly, responses to cannabinoids depend on ovarian hormones in rodents. And this has been seen in many studies. One of them, some of them include Dr. Cooper herself. Intact or sexually intact female rats, when compared to ovariectomized rats, which are rats that have had their ovaries removed or are hormone deficient, show greater self-administration of cannabinoid receptor type 1 agonists and actually develop greater THC dependence and exhibit greater withdrawal symptoms. So that goes to show that ovarian hormones play a role in sort of this cannabinoid signaling or reinforcing effects of cannabinoid. In addition to that, when rodents were compared across different phases of their estrus cycle, which on the right-hand side of this slide, you see the estrus cycle on top of the menstrual cycle, you see a similar peak in estrogen in proestrus or later proestrus. And this is thought to be somewhat equivalent or a proxy for the peak in estrogen you see in the late follicular phase in humans. And what preclinical studies found were that female rats actually exhibited greater THC-induced antinociception or pain-relieving effects in late proestrus when estrogen levels were relatively high. So this goes to show that during elevated estrogen levels, there was also an increase in sensitivity to these effects of THC. It was not just this whether the hormones there or not. It was the amount of hormones that were in the system circulating. Moving to the next important piece of preclinical evidence that I looked at is this very seminal paper that looked at estrogenic modulation of cannabinoid receptor density within the rodent brain. They actually tested different limbic areas, and I'm choosing to focus on the amygdala, given that one of the main effects that I was interested in was anxiety, and amygdala is thought to be one of the more emotional substrates of the brain. You can see in this figure that they compared 4 groups of animals between each other: male rats, female rats, both sexually intact, ovariectomized females, and ovariectomized females with supplemented estradiol. You can see here that both sexually intact female animals and ovariectomized female animals with supplemented estrogen had significantly greater cannabinoid receptor type 1 density within the amygdala, binding site density within the amygdala when compared to males or ovariectomized females, which suggests that estrogen or estradiol, the most prevalent form of estrogen during that time, during adult life or during adult animal life, was causing somewhat of an increase in CB1 receptor expression. It was having a positive effect, which made me think, and the authors of this paper actually, that this modulation, this estrogenic modulation, might affect or might underlie why women are more sensitive to the anxiogenic or aversive effects of cannabis and THC. So now that I touched on my preclinical evidence, I'm going to go into what main research questions I pulled out of this. First, 1, do circulating estradiol levels in humans impact physiological or subjective responses to THC in women? So this we can sort of just take on a basic level. We can administer acute doses to THC in women and see if their circulating estradiol levels impact this. And then the other one is, if we separate these women into early and late follicular phase, are we seeing a significant difference between how they respond to THC? And again, remember, early follicular is associated with relatively lower estrogen levels, while late follicular is more associated with the elevated estrogen levels prior to ovulation. So with these questions, I developed some hypotheses based on that preclinical evidence I showed you. 1, I hypothesized that variability in acute responses to THC would be significantly associated with circulating estradiols in these women, and that increased estradiol levels would lead to an increased sensitivity in the acute effects of THC. And then for my next research question, my hypothesis was that women would experience greater acute responses to THC during the late follicular phase than the early follicular phase, mostly associated to the increased estradiol levels during the late follicular phase. So with those research questions and hypotheses, I set off to develop a study. I'm going to go over my method now. I recruited women from the Chicagoland area, from the university and surrounding community, women who were not on hormonal birth control, and I randomly assigned them to 2 different groups, to the early follicular group and the late follicular group. All women within each of these groups had all of their sessions within that phase. I'll go into that in the next couple slides. And then all these women went through either 2 or 3 sessions. And this was because we wanted to look at dose response, but then we cut out the middle dose when we weren't seeing much of it. So we were just keeping the placebo in the 15 milligrams. And you can see here that, again, all 2 or 3 sessions were run in either the early follicular phase or the late follicular phase for these women. As mentioned, these were women between the ages of 18 and 35 years old. We controlled for BMI to somewhat control for absorption rate, no hormonal birth control users, and they all classified as occasional cannabis users, which we defined as having more than 4 times of cannabis use across their entire lifetime, but less than 11 occasions of use in the past 30 days. It's also important to mention that all individuals had to come in and provide a clean drug test on the day of each experimental session. They also were required to go through a physical and semi-structured psychological interview to ensure that they were in good physical and psychological health. And we did not include anyone with current or past moderate to severe substance use disorder. So what were the primary outcome measures? Some of them, well, first of all, the most important one for me was hormones, looking at both estrogen and progesterone. And during that time, I actually had the pleasure of learning phlebotomy, and I did most of my blood draws on all 60 women for all 3 or 2 sessions for them. It was a very fun learning experience. Then I also made sure to gather heart rate recordings, blood pressure recordings, and I attached them all to an ambulatory monitor to measure ECG signal. And this was just to get a more niche measure of cardiovascular cardiac activity. I also took saliva samples to measure salivary cortisol throughout the sessions. And then I used some very reliable and well-documented subjective drug effect questionnaires, including the drug effect questionnaire, the Addiction Research Center inventory, and the profile of mood states. So I'm going to take a moment to go into the high frequency heart rate variability, which was what I pulled out of the ECG signal I got from those ambulatory monitors, because I feel like some of you might not be totally familiar with this. I wasn't until I first started to take this head on. Essentially, we know that there is the sympathetic nervous system and the parasympathetic nervous system, and our parasympathetic nervous system is more involved with allostasis, or bringing our body back to homeostasis. Its main neurotransmitter of use is acetylcholine. And what we can do is, if we measure ECG signal and we measure heart rate variability, which is essentially the variability in interbeat intervals, so that's the time between each beat of the heart, we can fast Fourier transform that information, isolate a certain frequency range, and say, okay, this is specifically parasympathetic activation or activity. And so, as I mentioned, it's just taking these interbeat intervals, transforming them using respiratory information, which was also collected during the ECG, and isolating a certain speed of activation. And we can say confidently that that is the parasympathetic cardiac activation. Going back, now that I've touched on that a little bit, refresher, we have 60 women, 2 different groups, 30 women in each group. Women in each group had all 2 or 3 sessions run within either the early follicular or late follicular phase. They all came in, all sessions were run between 12 and 4 p.m. Upon arrival, they needed to provide clean drug, pregnancy, and ovulation test strips. So that was to ensure no drug was on board, none of them were pregnant, and none of them were ovulating. If they were ovulating, we would know that we had missed their follicular phase and we were too late, and we would have to reschedule them. We administered baseline questionnaires, took a baseline saliva sample, drew their blood, administered the ambulatory monitor, administered the drug, and then periodically until 4 p.m., we went in, had them fill out subjective questionnaires, took saliva samples, and recorded heart rate and blood pressure. Once we deemed them the drug effect to have worn off and ready to depart, we let them leave the lab. So to give you a little introduction to how I analyzed my data here, first, I analyzed it in regards to ovarian hormones. And when I say that, I'm talking about estrogen, progesterone, and then actually the ratio between progesterone and estrogen, because that's known to have clinical relevance. What I used here was a mixed effects regression model, and this was simply because it had the most statistical power and it could accommodate for, random missing data and for linear and quadratic effects across time. And this allowed me to put in time, both linear and quadratic effects, drug, and ovarian hormones as independent predictors. And I was able to check for the effect of drug across time, but then also the effect of ovarian hormones on drug effect across time, which is really cool. And then for the menstrual cycle phase analysis, I essentially did very similar analysis, but instead of using the ovarian hormone levels, I used menstrual cycle phase group, whether they were early follicular or late follicular women. So now that I've told you my entire experimental method, I'll move into my results. I'll start with the ovarian hormone, or I'll start with group characteristics and overall drug effects, excuse me. Here we see some group demographics of our two groups of women. You can see that they were not statistically different in age or body mass index. They also were not different in recent substance use, which was their alcohol, nicotine, and cannabis use within the past 30 days. And they also did not differ in their lifetime cannabis use either. The other measure I took at baseline was a trait anxiety inventory, since I was particularly interested in anxiety-inducing effects of cannabis, and they did not differ in their trait anxiety either. From our hormone analyses, we can see here mean estradiol levels by group. So for the 2 or 3 sessions each woman had, their estrogen or progesterone levels were averaged, and then they were compared between early and late follicular. And we see here a significant difference between our groups, in which late follicular women had significantly higher levels of estrogen, which is as expected and what we wanted. In addition, we see here with progesterone that there is not a significant difference between our two groups of women. We do see that there might be a slight increase, but it did not reach a statistical significance. And again, this is as expected given that image of the menstrual cycle and how progesterone stays relatively low across most of the follicular phase. Moving on to how THC affected the whole sample overall, I'm only going to touch on heart rate and a couple other measures, and then we're going to move into specifically how the ovarian hormone effects came into play. We see here a figure of heart rate across time, and the 3 different dose conditions are plotted. Blue is placebo, red is the 7.5 milligrams of THC, and green is the 15 milligrams. We're seeing a beautiful dose by time interaction at both doses, and we're seeing post hoc tests that at different time points, both doses are causing significant differences in heart rate from placebo. This is as expected and has been seen. This replicates many of the prior studies that have used acute THC administration. Now, moving on to that cardiac measure that I mentioned, that high frequency heart rate variability, what was very cool about this measure was we were able to show a dose-dependent effect, that it was decreasing across time in relation to dose. And that's cool because it's only been seen before in samples of men and smaller sample sizes in the past. So to be able to replicate this result in a group of 60 women was actually awesome. And that shows that we're seeing THC, oral THC, is essentially decreasing high frequency heart rate variability, which if you remember what I mentioned prior, that means that there is a decrease in parasympathetic activation as well. So essentially meaning that that nervous system that brings us back to allostasis, brings us back to homeostasis, that its exertion or activation is actually decreasing due to the drug across time. The last measure I want to show across the whole sample was salivary cortisol, another measure of sort of sympathetic nervous system activation. And this was somewhat interesting because when you look at male-female samples of how cannabis and THC affect salivary cortisol, the results are mixed. They're not always consistent. And when we see the results from this study, we see that first, the time course of salivary cortisol is much more delayed than any of the other physiological effects that we were looking at. In addition to that, we are seeing a dose-dependent response, and we're seeing the largest increase at the 15 milligram dose. This is very interesting, and personally would love to, extend our session lengths and maybe do a pharmacokinetic study on this, or looking across time, how this might play out in the descending limb or even the peaks of these effects. So overall acute effects of THC in our sample was it increased heart rate, it increased salivary cortisol, and this I didn't report on, but I'll touch on soon. It increased self-reported measures of feeling a drug effect, liking the drug effect, and feeling anxious. It also decreased high-frequency heart rate variability, which was very cool. It meant that THC seemed to decrease peripheral sympathetic activity while it increased peripheral sympathetic activity. And you can find more information on that in the paper below. Moving on to the other results, ovarian hormone analysis specific results. Is variability in response to THC associated with circulating estradiol levels? So first we see here across session, so session is on the x-axis, circulating estradiol levels. I showed you mean estradiol levels, but one thing I wanted to check was that estradiol levels did not change across session within individual. And we're seeing here that across all 3 sessions, late follicular women always had significantly higher levels of estrogen than late follicular women, or than early follicular women, excuse me. In addition, we see no significant difference in progesterone levels across all 2 or 3 sessions between our groups. What we found was that estradiol did not impact physiological drug effects, neither the heart rate, blood pressure, or the high frequency heart rate variability. And you can see here 3 different figures, one for each drug condition, placebo 7.5 and 15 milligram doses. Time is on the x-axis, y-axis is heart rate beats per minute. We are not seeing a significant difference between the 2 groups. And you're asking, why are these 2 groups, early follicular and late follicular? Well, the analyses were conducted using ovarian hormones as our primary predictor variable, but for ease, we're showing the figures in early and late follicular phase. So that's just to clarify any confusion about that. And then we also see that estradiol, although it didn't affect physiological drug effects, it does seem to have an effect on subjective drug experience. We see here DEQ ratings of want more of the drug, essentially a rating for craving or liking. And we see that both the 7.5 and at the 15 milligram, there is a more rapid increase in the onset or ascending limb of the drug effect in the early follicular phase women. Now, this is somewhat interesting because our hypothesis, or my hypothesis, was focused around increased estrogen levels in the late follicular phase would increase sensitivity. But what we're seeing here is that the women in early follicular phase, women with relatively lower estrogen levels, appear to be more sensitive to this ascending limb of the drug effect. And this was not only seen on this DEQ want more, it was also seen on profile of mood states anxiety. Now, we didn't really see a major increase in anxiety at the 7.5 milligram condition, but at the 15 milligram condition, we once again see this group difference, that the early follicular phase women, again, seem to experience the onset of these anxiogenic effects a little bit more rapidly or sooner than the late follicular women. So what can we draw from these results and conclusions? Well, one, self-reported ratings of subjective drug experience occurred slightly earlier in the early follicular phase, which meant that from our analyses, we could see circulating estradiol levels had a negative linear and positive quadratic effect on want more ratings, while it had a positive quadratic effect on anxiety scores across time. But when we looked at progesterone and the ratio between progesterone and estrogen, we did not see any of these, either of these predictor variables being related to any of the drug effects of THC across time. Moving on to the menstrual cycle phase analysis, not as exciting, but again, to introduce you to the question here, were the acute effects greater during the LF or late follicular phase? Now, Unfortunately, there were no further effects of menstrual cycle phase on the outcome measures, meaning that there were no phase differences that were not already accounted for by the ovarian hormone analysis. Essentially, neither the early follicular or late follicular group experienced greater peak physiological or subjective drug effects. So even though we are seeing some role of estradiol in time course of subjective drug effect, we did not see a difference in magnitude or peak drug effect in our early and late follicular groups. So what are our overall conclusions from this? Well, our main question was, is variability in response to THC associated with circulating hormone levels? And I hypothesized that increased circulating estrogen would be related to increased sensitivity. And we see here that most of the acute effects of THC were not actually related to circulating hormone levels, and it didn't increase sensitivity to acute effects of THC. But we're seeing that it plays a role in the ascending limb of a drug effect, which is definitely justification for further investigation. And then our second research question was, does acute responses to THC differ between the early and late follicular phase? And I hypothesized they would be greater during the late follicular phase. Well, we didn't see this. The only difference we saw was that subjective responses seemed to occur earlier in the early follicular phase, or in women that had relatively lower or depleted levels of estrogen. So where do I go from this, right? Well, first of all, replicate. We all know we want to replicate as much as we can. If I could do this study again with more time, more resources, I would do a within subjects design. Obviously, when dealing with menstrual cycle phase, there's so much individual variability that if we could have women come in and take the drug at every single different phase of their cycle with enough washout time, obviously, it would be best to compare a participant to themselves than to compare two separate groups of women. The other follow-up investigations I have in mind for this research would be a longer session design. You can see in some of our drug effects figures that although we caught the peak drug effect, we didn't capture the complete descending limb, which if we lengthened out our session design, we might be able to do sort of area under the curve analysis and compare the groups. As mentioned, the within subjects design, taking a stab at the luteal phase, that was the phase where we see that peak of progesterone in addition to the secondary peak of estrogen. That would be very cool to look into. And again, maybe the ratio between progesterone and estrogen might have something to do with this sensitivity. Another cool idea is using supplemented estrogen. So there have been studies done by Harriet with stimulants where they had women have a patch, an estrogen patch, and they looked at whether that affected responses to stimulants. So that's another avenue to explore. And the estrogenic modulation paper that I used to justify my human work also touched on how cannabinoid receptor density is modulated by estrogen in both the hypothalamus and the hippocampus. So looking at different behavioral tasks under the influence of THC might be interesting. And then more sort of dream exploratory investigations would be somehow scraping together the funds to do a PET study of the CB1 receptors across the menstrual cycle. And then, some of the work that Dr. Cooper has been doing by checking into other phytocannabinoids or terpenes that play a role in whole plant cannabis and other routes of administration, right? I used oral THC here, but marijuana and cannabis, it can be commonly smoked. So seeing if the difference between smoked or oral might play a role in these hormone or sex-related differences. And something to mention is that even though there's not a lot of research based on why, cannabis should be used for menstrual cycle pain, it is still being advertised to a lot of women. And there are a lot of women who are still using it to cope with menstrual cycle pain. And this recent 2015 study actually surveyed 192 women and found that 85% of them said that they use cannabis for menstrual pain, and that almost 90% said it was effective at relieving pain. So even though the research isn't there, the people are still going to be using. And that's why it's really important we continue this line of research. We get to understand these effects. We understand how female and hormones across the menstrual cycle affect this, so we can better guide both medicinal and recreational cannabis users towards safer cannabis use guidelines. And with that, I'd like to thank everyone who was involved in this work, particularly my advisor, Dr. DeWitt. She has been an invaluable mentor, as well as my co-sponsor, Dr. Norman. Dr. Hedeker helped me with my stats. I would have never been able to do those analyses without him. CC was so generous to do the hormone analyses with the help of Paul Rue, the rest of my thesis committee, all the rest of the human behavioral pharmacology lab, and then, of course, my funding sources, and UCLA and Dr. Cooper for inviting me today to give my talk. Thank you.
That was great, Dr. Pabon. Thank you so much. So opening up the floor for questions, please feel free to either put your questions in the chat box, or since this is a seminar style, feel free to raise your hand and we can just have a conversation. But I will start, if it's okay. I had a question in the chat box. So I just had a question. It was interesting how the two subjective ratings that you saw differences in the women who were tested in the earlier follicular versus the late follicular phase were kind of contradictory, right? You had increased ratings of want more drug. So that kind of indicates that it produces a feeling that is positive, but then you also saw an increase in ratings of anxious. So I'm wondering if maybe the women who were endorsing higher ratings of want more drug were actually different than the ones who were rating higher ratings of anxious. And then I was also wondering if maybe they have different levels of estradiol during their early follicular phase. I don't know if you had a chance to probe these like very fine-tuned individual differences, but that was what first came to mind.
Yeah, so I had a very similar question when I saw that it was want more and anxiogenic. It didn't make complete sense to me. So I actually looked at the individuals, and most individuals who experienced the anxiogenic effects did not want more of the substance. So you have some rationale behind that something sort of distinguished these women, the women who experienced more anxiety and the women who experienced greater wanting of the effects. And we have the data to look within the late follicular phase and look at the estrogen levels of these women. I wonder if it's, I don't know if because want more is slightly different of a self-report subjective measure than, a mood state, whether that is something to consider, but definitely an interesting area to look into, because we did see variability in the late follicular phase women in their estrogen levels. So specifically looking at average estrogen levels in the women who, I haven't done that specific analysis, but I will, because that's very interesting.
Great. Thank you. And then we have a question from Dr. Murray and then Dr. Gelberg. So Dr. Murray, if you want to just chime in rather than reading from the text box.
Yeah, so I was just thinking more about what want more really means, and thinking about two people, going to a bar and they take a few shots of tequila and one person's just like, I cannot have any more. Like, I am so intoxicated. I'm already way too drunk. And someone else is like, no, I could do a little more. I'm hardly even feeling this, so whether or not want more means they're less sensitive to THC in this case, and whether that would be in line with your original hypothesis.
Yeah, I think that's a fantastic point, and just what I was sort of getting at, right? Want more is this subjective self-report measuring of a craving or like liking a drug. And depending on line of thinking, that might be because a person isn't as sensitive to a drug, but it might also be because a person has heightened sensitivity to a drug and they want it as well. So I think it's difficult to really distinguish what's happening there, but it could definitely be in the direction I want it to be.
Cool, thanks.
Great, thank you. So we have Dr. Gelberg and then Megan Massa. Dr. Gelberg, are you able to speak?
Yes, I am. Thank you so much. I really said it in my chat. Excuse me. It's wonderful to see your work in women's health and in focusing on this. I mean, you have a career where you could be looking at all of the drug du jour on its impact, and it's complicated, and women are complicated and very interesting. And I was thinking about our work. We're screening UCLA primary care patients for their cannabis and tobacco use and secondhand smoke and medical cannabis reasons. And many of them report that they're using it for menstrual pain and pelvic pain. So it's nice to see sort of the health services and the, bench science working together to support each other. So thank you. And you've got a great career ahead of you.
Thank you so much. I appreciate that.
Thank you, Dr. Gelberg. It's great to see you here. And Megan Massa.
Hi, thank you so much for that talk. It was really interesting. I'm also wondering, based on a previous question, as to whether your want more isn't necessarily a measure of liking, but is a measure of motivation, right? Because estradiol can really impact the motivation centers of the brain. It doesn't necessarily mean they like the drug. So I was wondering if you had any thoughts on that and the differentiation between liking and wanting.
I think that's a very interesting question. We included DEQ like as one of our measures, and we did not see a difference in or an effect of estrogen on the like ratings. So because we had a like specific rating, it makes me think that you're right. It might not necessarily be a proxy for liking, but more so drug seeking. And so that would be an area to explore. One area I want to look into is impulsivity, because hormones play a huge role in impulsive action. So seeing if these women across the menstrual cycle have changes in impulsive action under the influence of THC might be another sort of avenue to explore for that. But very, very important point to make. Thank you.
Thank you.
Thank you. Dr. Muller, it's great to see you.
Hi, first off, really fantastic presentation, really appreciated it. I'm an endocrinologist, so this was very interesting to me. I really loved how you characterized early follicular, late follicular, and looking at the behavioral changes. I'm wondering about how we could address the challenge of kind of translating this into, how people use, right? People who use cannabis, even moderate users per se, right? There are hypothalamic and pituitary changes to the gonadal axis with downregulation of CB1 receptors and et cetera. So how can we account for some of the impact of that when you're thinking about trying to study this long term? Because most patients or most women who might use are likely not going to use one and done, right? There might be some habitual factors. So how can we evaluate that, I guess, is my question.
That's a fantastic question. And yes, there is definitely a bunch of literature out there that shows that chronic cannabis and cannabinoid use can have lasting endocrinological effects on the ovarian hormone levels and also on testosterone levels in men. So I think that's a very relevant question. And, if if there was a better understanding of the chronic effects of cannabis use on these hormone levels, maybe we could use that as a basis for each individual. We could use their use history, their frequency of use, how often, how many years they've been using, and sort of equate that, okay, how much has their endocrinology shifted due to their chronic use, and how is that going to change the effect? So, it would require a whole series of studies in which we look at people who have different histories of use and who use the drug at different frequency rates, because then we would be able to see, would frequency of use, would chronic use affect these effects we're seeing? And so it's very, very interesting and very pertinent. You make a huge point. It's hard, to do these types of questions in controlled lab environments, but that would be a next step for sure.
Great. Any other questions? I just have one more to ask. This is an obvious question, Elisa, coming from me, who studies a lot of people who use cannabis daily. I'm wondering if you have hypotheses with respect to what you would expect to see in the women that you've looked at here, who are very light users. I mean, they've used 25 times in their lifetime, versus people who are using, more heavily, several times a week. Do you expect to see differences as a function of estradiol levels, or do you think that there's just an overall effect of estradiol increasing sensitivity to THC effects?
I think it definitely depends on the use of the individual, because use of cannabinoids in cannabis can have major modulatory effects on cannabinoid receptors within an individual in itself. So even if estrogen is playing a role in modulating cannabinoid receptor density, use of cannabinoids itself is going to affect it. So it would be a question of looking at this how estrogen affects, different users, right? So like I said, these are light users. Maybe we explore this question in a heavy use group with people who use every day. But there's the idea of tolerance, why we would need to develop and have, good measures to determine what would be sensitive in this group of chronic or daily users. But yeah, I definitely think that's interesting.
Good. Dr. Peterson.
Thanks for your talk. It was really interesting. I'm interested personally in ovarian hormones and how they affect cigarette smoking. And I have to admit, I'm like a low-key unsurprised by the finding that estrogen doesn't facilitate drug taking in the way that it does in animals, because I did a project with a similar sort of null finding where we measured dopamine receptor density over the menstrual cycle and saw really no change. But I guess my question then is, I'm wondering, I'm just unfamiliar with the cannabis literature, and I'm wondering, so this idea that you and that Connor had mentioned, that expressing a lower, I think it was lower wanting or lower, it could be that they're satisfied, or it could be that they're less interested. It could go either way. So I'm wondering if there is literature just evaluating the rate of ad lib drug taking over the cycle. Do we know if women at different phases just use less or more?
There's a very old study from the 80s. I think it's either Lex et al. Or Griffin et al., where they surveyed a bunch of women and they asked women what phase they were in and how much they were using, and then whether their use changed across phases. And they didn't find anything.
Well, we're collecting some data in our lab, so we'll be able to look at. We have women filling out daily rating forms every day across their menstrual cycle phase, so we might be able to dig into that a little bit. Which will be interesting.
Yes. Yeah. Obviously, these are old studies. We have much more advanced methods and larger sample sizes, so I have confidence that we'll be able to look deeper into that.
Great. With that, I'd like to thank everybody so much for coming. And of course, to Dr. Pabon, thank you so much. It was lovely to hear about your work and your interests. And we're looking forward to seeing you in person one of these days, hopefully, as convenient as Zoom sessions are, it's always good to see people in person. Thank you guys so much. And we look forward to seeing you next month on April 28th for our faculty meeting. Take care.
Thank you so much.
Thanks, Elisa.
Bye everyone.